Thank you. Good morning to everybody. Welcome back to the second day of the course. I hope you had fun last night. I would like to ask how many of you went back to Chicago and came back this morning to have some fun? None? Okay. That's good because this morning is a full morning. We are talking about fluorescence. What is fluorescence? Fluorescence is the emission of light that a substance has upon absorption of light. Here I have some tonic water, and I brought it because Professor Clegg, I know he likes it, but I brought it also for another reason. I have a UV lamp. If I put the tonic water close to the UV lamp, you see light it up. Why? Inside there is some quinine. The quinine absorbs some radiation emitted by the lamp in the UV. The fluorescence is this bluish light that you see with your eyes. So the question is, what do we do with fluorescence? Why is it important? How does it help us in our research? So we will explore a few opportunities this morning, and we just touched the basic of fluorescence. Next week you will have a chance to look at some instruments. Here are some fluorophores. Typically, a lot of the aromatic molecules emit fluorescence, but fluorescence is also emitted by some minerals, some salts, like uranium salts. And the probes are used, you see the famous quinine from the quinine sulfides. The probes are used also as a medium to attach to other molecules which are not fluorescent or are slightly fluorescent. Well, fluorescence became important in the last few years, partly because the availability of lasers, the availability of chemistry probes, and the availability of instrumentation. Why is it important? It is important because we can use it to understand the environment of molecules. We can use it to understand how some processes occur on a very short time scale, all the way from the microsecond all the way down to the picosecond scales. Several parameters affect the fluorescence. Here, I just list a few. So if we place a fluorescent probe into a medium, the fluorescent probe characteristics are influenced by the status of the environment, be the pH, be an electrical field, be the polarity. Recently, with the availability of multi-photon lasers, we've been able to investigate what happens inside the cell. Well, there are several processes that are very interesting. For instance, there are some proteins that cross the membrane, goes inside the cell to specific locations. Using fluorescence, using the instrumentations, we can actually follow the path of these proteins, see where they go within the cell, which location, do they cluster, do they form dimers, trimers inside the cell. So this is another powerful tool that we have at disposal in order to analyze these complex systems. In the past few years, it has become routine to measure the single molecule detection. Fluorescence is in fact very, very sensitive, and is practically routine today to be able to go down to a single molecule. Now, these are some of the examples of structures where fluorescence is used. Of course, we have molecular dynamics. It can be the interaction of proteins. It can be nucleic acids to see what's happening, the distances between different fluorophores. Cell studies. We engineer some animals using green fluorescence proteins, and we can acquire images of these animals that glow when exposed to light. And of course, fluorescence is used in a variety of instances to do microarrays, to study new drugs using microwave plate readers. There is a variety of instruments, gadgets, that are used to carry out all the different measurements of fluorescence. This is a classic fluorometer that you will see next week during the course, during the practical in the laboratory. It's a confocal microscope. We have a flow cytometry. This is made by Eyesight, a local company. Fluorescence is used in genome sequencing, and this is a specific machine made by Pacific Biosciences, and immunoassay chemistry. about diagnostics. There are thousands of these instruments in the hospitals all over the world. Microwave plate reader used essentially by pharmaceutical company during the drug discovery process. Well, let's talk a little bit about the history of fluorescence, and I just touch a few concepts to understand where it comes, how we got to the knowledge that we have today. The first reports of fluorescence were recorded by this fellow, Monardes. He was a Spanish physician, and he wrote an extended encyclopedia talking about the discoveries in the New World. Although he never visited the New World, America, he was collecting all the information that the sailors and other explorers were bringing. Specifically, he reported that there was a wood that, when placed in water, would have released some bluish color, and that was the first report of fluorescence. This phenomenon was popularized in Europe by Charles de Lecluse, and he gave to this particular piece of wood the name Lignum nephriticum. And for some reason, they thought that it helped in treating kidney diseases. In fact, an Englishman, John Frampton, who lived for several years in Spain and then returned to England, also translated the Monardes' work in English, and these were his observations about the wood. It's interesting to note that even Robert Boyle, the famous Robert Boyle of the pressure-volume gas law, was interested in the phenomenon, and he made some interesting observation. By making the solution more acidic or less acidic, the fluorescence increased or decreased. And above all, he noticed that if he used this piece of wood several times over the course of an experiment, the intensity of the fluorescence would decrease. In the next couple of hundred years, people used practically this lignum nephriticum for medicinal purposes. But people didn't understand what's happening. And only recently, only about 10 years ago, Professor Ulises Acuña in Madrid came up with an explanation and was able to isolate the compound that gives rise to fluorescence. In the same years, at the beginning of the 1800s, people were very interested in the quinine sulfate. Again, it's the same substance in a different forms that we have in the tonic water. Why was that so important? Because quinine sulfate was used to fight malaria. England was sending explorers, soldiers all over the world, and they were sick, and that was the only way to cure it. And so the scientists became interested in this substance, and Herschel, the great astronomer, discovered the fluorescence from the quinine sulfate. Of course, at the time, the word fluorescence wasn't really used yet, and he called it epipolyc dispersion. So I wonder if we can go in a lab today and just say, can you acquire an epipolyc dispersion of quinine sulfate? Okay. And this is his paper that he published back in 1945, talking about the phenomenon. After him, the phenomenon attracted the attention of Stokes. Stokes was a professor in Cambridge, and he came up with a very interesting experiment. He put some of the quinine sulfate solution into a bottle, into a vial. The problem is that he didn't have the UV lamp, and he came up with an ingenious experiment. He used white light from the sun. He dispersed the white light through a prism so that the different wavelengths of the light could be dispersed, starting from the UV to the red. He moved the vial in the different position. It was a small vial, it wasn't a big bottle. And finally, when he was in the region of the near UV, he saw that the fluorescence, the quinine sulfate, was emitted this light. And he made this kind of interesting comments. He published a paper at the time, more than 100 pages long, and to describe this phenomenon. And initially, he called it dispersive reflection. Although he also wrote a note where he said, well, I don't quite like this name, And so he proposed the use of the name fluorescence. And after that, the terms stayed being used throughout the years. He made also some interesting observation. One was the Stokes shift. He noticed that the fluorescence light is emitted at a wavelength that is longer than the excitation light. And later on, a few years later, he also suggested that fluorescence could be used to build some instrumentation to detect, in this case, organic substances. It is interesting, the story of the quinine sulfate, because this substance remained important in the history. And during the last World War II, when the war was going on in the Pacific and the routes, the sources of quinine were blocked, the American government was very concerned because of the soldiers again going out and because of the malaria. And so the government started a lab close to Washington, D.C., where they were studying the amount of drugs that were in the bloodstream of people. And this was built by these two researchers, Brody and Underfriend, and that practically was the first fluorometer. It's interesting that after the war, the government was expanded, and this was the government that eventually became the National Institutes of Health, which plays an important role in our daily life. Let's go back to the history of fluorescence. So, the modern fluorescence really started with the beginning of the quantum mechanics. So in the first 30, 40 years of the last century, several phenomena were understood about the fluorescence. And after that, we use still a lot of these phenomena, of this understanding, to explain what's happening today. Okay, let's go back to business. What do we measure about fluorescence? In fluorescence, there are basically 5 parameters to be measured: the excitation and emission spectrum, the quantum yield, the lifetime, the decay time of fluorescence, and the polarization or anisotropy. So we will touch all of these 5 parameters briefly in the following. So to start, we start with a quantum mechanic representation of the levels of a molecule. So the molecules can be represented by a ground state and first excited states, other excited states. The absorption, and with each state there are vibrational levels associated. The absorption happens typically from the ground state to the first states or to upper states. Upon absorption, the energy trickles down to the lower level of the first excited states, and from there the light is emitted, and this is the fluorescence. Is emitted with a rate constant Kr. Some light is emitted to some high vibrational states associated with the first ground states, other with lower. So this is the typical phenomenon. This is not the entire story. In a molecule there are also so-called triplet states, which have a different spin multiplicity. So some of the energy trickles down to the triplet states, and from there it's emitted from the lower triplet states down. And this is typically called phosphorescence. There are also other ways of releasing the energy, and these are non-radiative processes. There is an intercrossing system between single state, triplet states. There is a decay, a radiationless decay within the same singlet states. So basically this explains what's happening. The typical times associated with the processes: the absorption is very fast, of the order of 10 to the minus 15 seconds. The fluorescence is of the order of 1 nanoseconds, but it can go from the picoseconds all the way to almost the microseconds. Phosphorescence is typically above 1 microsecond. So 1 microsecond, it can last even seconds. Now, let's talk about an excitation spectrum. So what happens with the excitation spectrum? To record an excitation spectrum, we want to see what are the what is the range of frequencies or wavelengths or energy that a molecule absorbs. So in a spectrofluorometer, what we do, we position the spectrofluorometer at an emission wavelength, and so let's say a certain wavelength that we pick up into this region, and we vary the wavelengths of the excitation, and we record the intensity of the emission. This is a typical excitation spectrum, rose Bengal, which is a dye that is used also in medicine, in eye cure. And so what we can see here is that the emission is acquired above 600, and we scan from 400 nanometers to 600, and it has this shape. So the shape is dictated by the way the vibrational levels are associated with the specific single level of the molecules. Now, when you use an instrument, typically you don't acquire what is called an absolute excitation spectrum, but there are some corrections to be done. I don't go into that. We will talk during the demo next week. And all I can say is that usually manufacturers provide these corrections to the data so that you can correct your data. Similarly, for the emission spectrum. So in the emission spectrum, one fixes the excitation wavelengths and then scan the emissions from one wavelength to another. So going back to the Peranger-Jablonski diagram, so we fix the excitation at a certain level, at a certain wavelength, and then we go through the emission and we scan we acquire different photons associated with this decay. And this is what it looks like. Of course, we can superimpose the excitation and the emission, and from here we understand, by looking at the Peranger-Jablonski diagram, the Stock shift that was already noticed by Stock. Then we also noticed that the fluorescence spectrum, it looks very similar. It looks like a mirror image of the excitation spectrum. And this goes back to the characteristic of the Peranger-Jablonski diagram. It is not true for all the substances. It is not true, especially some substances in the excited states change the conformation. But the rule is followed by several substances. What is also important to notice is that the emission spectrum doesn't really change if we excite within the excitation spectrum different wavelengths. In other words, if we were to excite at this point about 520, or at 500, we still would get the same emission spectrum. Now, back to the third parameter, the quantum yield. The quantum yield is defined as the number of photons emitted in a radiative way, photons that we see, divided by the total number of photons absorbed. Why it is important? It is important because it's different for different substances. So here we find the quinine sulfate, and we see that almost 55% of the photons are emitted, which is pretty good. But there are some substances where the quantum yield is very low. So that means if I want to show fluorescence with benzene, well, it's not a good choice because only 5% of the total photons absorbed are re-emitted. On the other hand, there are some other substances. Rhodamine 101 is 1%, 100% of the photons are emitted. This is important if I want to use one of these probes as a marker for some molecules that are poorly fluorescent. So I need to pick up the proper molecule. Of course, there are also other considerations, how this molecule attaches to the other molecules. But the first parameter to look is really the quantum yield of the molecule. Now, another very important parameter is the decay time or lifetime. So how do we introduce this concept? So we have the ground level as 0. We have the first excited level as 1. So suppose that we have a solution with fluorophores, and we send a short pulse of light to the solution. So some of the molecules from the ground states finish up in the first excited state. At that point, suppose that the population is N1, the population of the first excited state. They decay with a rate constant. They decay radiatively, and there are some processes, some routes, which are not radiated. Yet, the population, the change of population in the first excited states follows this equation. We can make Ft equals 0 if we start the origin of the time. If we integrate the equation, we simply measure the population of the first excited state at any time t as the initial population time an exponential minus t over tau s. Tau s is this number, and tau s is the lifetime of the first excited level. This is the definition. In other words, we can even say that if we have a population of molecule at time 0, the lifetime is when the population decreases by 1 over e, so about 37%. Using this definition, we can recorrect the definition of the quantum yield as the ratio of the lifetime of the first level divided by the radiative lifetime. Now, the lifetimes, in principle, can be calculated using this Strickler-Berg equation. By the way, this is a 8 pi times 230, so the symbol is a multiplication. The equation is fairly complicated. It doesn't work most of the time because of the complexity of the system. And in fact, if we look at, for instance, some aromatic molecules, we can see, like going from 2 rings, 3 rings, how the lifetime changes. Yet there is no clear pattern to predict how the lifetime will go. For instance, we are talking about a few nanoseconds here, and in this case, pyrene, we're talking about 400 nanoseconds. Similarly, the lifetime, and this is the most important aspect of this parameter, is highly influenced by the environment, by where the molecule is in. These are some examples. This is ANS bound to a protein. ANS in water is about 100 picoseconds. Yet, when ANS is bound to a protein, let's say BSA, the lifetime can be 8 to 10 nanoseconds. So it changes order of magnitudes. Ethidium bromide in water, 1.8 nanoseconds, bound to DNA, 22 nanoseconds, tRNA, 27 nanoseconds. So we can use these observations to carry out a lot of studies. The lifetime of tryptophan. Tryptophan is one of the amino acids in a protein, and one of the two, basically tryptophan and tyrosine are the ones that emit most fluorescence. Yet in proteins, it ranges from about 100 picoseconds to about 10 nanoseconds, because it depends where it is located in the protein. Is it outside, in contact with the solution? Is it inside and protected by the environment of the protein? There are two ways to measure lifetimes. One is time domain, the other is frequency domain. Time domain is the more straightforward. We take a short pulse of light, ideally is as short as we can, although practically it's not short, nothing is short. We send this pulse on the solution, and we detect the photons that come out from the solution at different time. And so for a single exponential decay, we find this expression, we fit it, and we determine the decay time. In frequency domain, the light beam is CW, so it's continuous, but it's modulated. So if we were to take a picture of the light beam, it will be a sinusoidal wave, and it's modulated at a frequency f. The emission is still modulated, but there are two important characteristics of the emission. One is that the emission is phase shifted. You see that the peak don't coincide, but the emission starts later on. In fact, some of the photons stay in the excited states. The emission, the peak to valley amplitude, is, which is called the modulation, is less than the peak to valley amplitude of the excitation. So we measure these two parameters, the phase shift, the angle, and the ratio of the peak to valley amplitude. And both are related to the lifetime using these formulas. And this is for a single exponential decay. Now, this is a representation of the time domain. So in time domain, we have pulses coming from the laser, one pulse, another pulse, and they continue. And there is a photon coming, and the instruments record the time between one photon and the next pulse coming from the laser. We may take this time, we build it, we place it here. So on. The second photon is a different time, shorter, it will finish up here. The third photon is in the middle, it will finish up here. We collect about 5,000, 10,000 photons, and so we finish up with this curve. And then we fit the curve and determine the decay time. In analog frequency domain, we use synthesizer to produce this modulation of the light source. There is a second synthesizer that produces a modulation similar to the initial modulation. It goes to the light detector. This allows to determine the phase shift and the demodulation. And this is the typical results. This is called the Weber plot by the late Professor Weber, who introduced it. So here we see how the phase shift changes with the increase in the modulation of the signal and how the demodulation decreases. To understand these curves, one has to remember these equations. So the phase shift increases with the modulation frequencies, which is out here, and the modulation decreases, well, we need to bring it out, with the frequency. Now, ISS offers both type of methodologies, and this is an example of an instrument that you will see next week. I don't go into the details now. I just want to show some of the results that we get. So using a time domain, this is NATA, is a tryptophan derivative. We have the narrow pulse, which is not so narrow, of the LED, pulse LED. We measure the decay, we feed the data, and we measure 3 nanoseconds. The same compound in frequency domain, we measure 3.1 nanoseconds, while the filter is slightly different, and even the excitation wavelength is slightly different. This is an example of a short decay time. This is the Rose Bengal. We look at the excitation and emission spectra. In time domain, we measure 77 picoseconds. In frequency domain, 78 picoseconds. I just wanted to give you an idea of how short the decay time can be measured using the current instrumentation that you may find in your laboratory. This is an example of time-resolved anisotropy. I will talk later on about that, the meaning of that, but again, it's coumarin 6. I just want to point out the decay times. So the lifetime is 2.3 nanoseconds using time domain, 2.33 frequency domain. The rotational correlation time, and I will introduce the concept later on, is 2.6 and 2.0. By the way, this is different because the samples were measured at different temperatures. And we will get back to that later on. We'll skip the analysis. Some applications. This is an application that requires the concept of FRET, forced resonance energy transfer. We won't go into the details. I just want to show how the concept is used. So we use a molecule that we call donor, and there is a molecule that is called acceptor. What happens is that if the donor gets close to the acceptor, the decay time of the donor decreases. So we can use this phenomenon to understand a lot of what's going on in specific instances. For instance, one typical application is immunoassay, where we have antibody and antigens. So antibody are marked, antigens are marked with fluorescence. In this case, there is energy transfer. So the decay time of this antigen, of the marker of the antigen, it's not the antigen, it's the marker of the antigen, is less than the decay time of the antigen when it's alone, because part of the energy is transferred to the donor, to the acceptor. This is yet another application of lifetime measurements, is a sensor. So the idea here is if we can use a molecule whose lifetime is affected by the environment, we can use that molecule to monitor how the environment goes. So we build a sensor. A sensor is, again, it depends on the analyte that we want to acquire. It can be also, it can be a specific molecule, it can be oxygen in water. So if we build a sensor, a chemical whose lifetime is dependent upon the concentration of the oxygen, then we can use this concept to measure the concentration of oxygen in water. This is an application made by Professor Thompson, who is at the University of Maryland in Baltimore. He devised some chemicals whose lifetime is dependent upon the concentration of copper ions in seawater. This is the difficult part, it's not the instrumentation. Seawater is full of a variety of molecules, so he was able to devise a specific molecule that was sensitive to this copper ion. He did a calibration, and using a frequency domain approach, he measured how the phase angle changes, the phase angle of that specific compounds in presence of different concentration of copper ion. And this is the molecule. So now what he did, he used this vessel, he carried the instruments on the vessel, this is a cartoon of it, and he merrily went out in front of the Atlantic. He used a fiber optic up to 250 meter longs, and he monitored the concentration of copper ion at different depths in the Atlantic Ocean. This is, again, I just want to give you an example of an application of lifetime, how people use the lifetime, this concept. Now we are left with the polarization. The polarization is another one of these properties of the fluorescence, and it requires some attention because there are several steps in order to understand how to use it. So light is an electromagnetic wave similar to standard radio waves. It propagates with, it has a wavelength. It propagates with a speed that is the speed of light. And there is an electrical field and a magnetic field which are perpendicular to each other and perpendicular to the direction of propagation. We will be concerned only with the electrical field, so the vibration of the electrical field in time. On the contrary, natural light is a superposition of several of these wavelengths of different polarized light. So we can envision, we can describe natural light as the superposition of a wave where the electrical field vibrates in a direction, in a different direction, makes another angle. So if we were to look from along the axis of propagation, we can indicate unpolarized light in this way. Now, there are some gadgets called polarizers that allow to select a plane of polarization in the unpolarized light. In other words, I have unpolarized light, I pass it through polarizers, and I get plain polarized light. Now, these polarizers are made by, there are the Polaroid type, and these are what are typically used in sunglasses, or a calcite polarizer, and this is a mineral that is birefringent and it disperses the two planes of polarization. Now, let's think of how to use the polarization with molecules. And this is the concept of photoselection. If we look at this aromatic, this is anthracene, we see that the anthracene goes to the first excited level with what is called a transition moment that has this direction with respect to the plane of the molecules. There is another transition moment that sends the molecules to S2, which is along the long axis of the molecule. So each molecule absorbs light according to the orientation of the transition moments. Now, if we have a solution with several molecules, they are all oriented casually, randomly within the solution. If we excite the solution with vertically polarized light, we only select the molecules whose transition moments are more or less oriented along the direction of the polarized light. In other words, let's say that we use vertically polarized light, and I have a molecule whose transition moment for absorption is oriented perpendicularly. There is no absorption, so the molecule doesn't absorb the light. On the other hand, if the transition moment is oriented parallel, there is maximum absorption. If the transition moment makes an angle, it goes as the cosine square of the angle. This is photo-selection. So out of an ensemble of molecules, we only select a few molecules within the ensemble, and we select the molecules whose transition moments are more or less oriented according to the direction of the polarization of the incident light beam. An experiment of polarization is typically run in this way. Here we have our cuvette, where we have our tonic water. We have the light source. We have the polarizer that selects the polarization of the light, vertical. We acquire the fluorescence, and we have a polarizer. And we can orient the polarizer in two different directions, the vertical or the horizontal. Or we can even call it the parallel to the z-axis, the axis of symmetry, or perpendicular to the axis. So this is the schematics of the experimental apparatus that allows us to measure the polarization. And we define the polarization as the amount of light that is parallel. This symbol is meant to be parallel, minus the amount of light that is perpendicular, divided by the sum. There is a different concept that people use, but it's exactly the same, and that is called anisotropy. The difference between the two is in the denominator. In one case, we have a 1 here. In this case, we have 2. What is the difference? If we look at the axis of emission, what we have with the anisotropy, we divide by the total intensity emitted. And the two concepts are used differently. In the life science, medicine, people prefer using the polarization. In biophysics, chemistry, people prefer using the anisotropy. The reason being that a lot of the formulas get simplified when using the anisotropy. Now, these are the limits of the value of the anisotropy and the limits of the value of the polarization. Well, practically, this never happens for molecules. Why? Well, there are several reasons for it. And we need to go back to the orientation of the transition moment for the absorption. So let's say that again that we excite a population of molecules at time 0 by a short pulse. And the moments, the absorption moments, are oriented at this angle, making an angle theta with the z-axis. This is our excitation, the electrical vector, and this is the z-axis. If we do the calculations, this is again parallel minus perpendicular, we come out with this relationship. And this is, if we use this relationship and we see the relationship between the emission and the absorption, and calculate the average, we find 3/5. And if we plug it into the previous equation, we found R0, 0.4. So this is the maximum value that the anisotropy can reach in molecules. It's also called the fundamental anisotropy. When doing experiments, we rarely measure 0.4 because molecules are getting depolarized. Also because this assumes that there is no motion of the molecule. But if there is a little molecule, we find a value that is lower than 0.4. And typically, the value that we measure is called the limiting anisotropy. Now, what happens if the moments of absorption and emissions are not parallel, but make an angle, alpha, between the two of them? The anisotropy, the R0, the fundamental anisotropy, has a term that is similar to what we found before, and a second term that has the same shape, but with the angle between the two moments. Now, we know the first value, which is 2/5, it comes from this example, and we are left with this other value. Now, if the two moments are parallel, we find again 0.4, as in the previous case. If they are perpendicular, we find minus 0.2. Now, the two values correspond to moments which are parallel and perpendicular. So if we go back to look at anthracene, we see that in this case we have two absorption moments which are perpendicular one to the other, and one sends to the first excited level and the second to the second excited level. So typically the value minus 0.2 or negative is associated with transition that involve higher levels higher than the S1, the first excited level of the molecule. Now, let's see how practically this plays a role. So here we have erythrosine, which is this molecule. We have an absorption, an excitation spectrum. So this is again the wavelengths where these compounds absorb light. Here we measure the polarization at different values. So it's called an excitation polarization value. You see that it starts at minus 0.1 at these wavelengths. So most likely here we are going to probe levels higher, the S1, the first excited level. And then the polarization tapers off to this particular value close to 0.3 and stays there throughout the point. Now, how to use all of this concept, this information? Polarization, molecules get depolarized. Why they get depolarized? There are several reasons. One is because molecules are not alone in a solution, there are other molecules. The main effect is the Brownian motion. So molecules get in collisions with other molecules of the solution. So once the molecule absorbs some light and has a moment, let's say, that is parallel to the excitation field, it gets in collisions with other molecules, and during the lifetime it might rotate. And so now we measure a different value of the polarization. So if we still use a very narrow pulse of light, we can characterize the decay of anisotropy using this formula. If we assume that the molecule is a spherical molecule, we find this expression when here we have D, and D is the diffusion time that is related to this equation that is called the Stokes-Einstein equation. There are two ways to measure D. One, time resolved. We can do the measurements, but we can also do a steady state polarization measurements by seeing what happens to the polarization by doing this integral. And these equations was first derived by Perrin. And it's a very elegant equation because it shows how the anisotropy goes with respect to the limiting anisotropy, to the decay time of the molecule, and to the rotational correlation time, this tau C being 1/6D, the diffusion time. In fact, we can use this equation to estimate the decay time of molecules, even if we don't have an instrument to do it. Let's say that we're looking at a globular protein where we, like BSA, and we attach a probe like ANS. We first calculate the rotational correlation time. So using the previous equation, we have here the viscosity. Let's go back. So in this equation, we have R, the constant of the gases, temperature, volume of the molecule, viscosity of the solution. We can replace the volume by the mass and the specific weight. For the viscosity at room temperature, we use 0.01 poise, 66,000 Daltons. We have 0.73 plus, this is called the hydration factor because the molecule, the protein, gets some water. RT, we do all the calculation, this should be equal. So we find 28 nanoseconds. From the steady state measurements, we measure a limiting anisotropy, 0.36, 0.23 for the anisotropy. We do all the calculation, and again, this is a times, is a multiplication, is 35, and we measure about 20 nanoseconds. So this is just an estimate based on steady state measurements. That shows to you how powerful this relationship is. Now, most of the molecules are not spherical. They are they can be described as ellipsoid, or they can be, and in that case, the anisotropy has a more complicated expressions. Some other molecules are hindered. So if we have a molecule that is placed in an environment where the motion is limited, we call it a hindered rotators, and there is a more complicated expression. All these are used to, in several fields, to study, for instance, in molecular biology, the situation in biological membranes, micellar systems, and here you see a few of the application of polarization. So what we did here, we touch, this concludes my talk. So we touch the fluorescence. We talk about the 5 parameters that are measured with fluorescence: excitation, emission spectra, lifetime, quantum yield, and polarization. Next week, during the laboratory, you will see how to measure practically a few of these parameters. The next lecture is by Professor Clegg, and he will go more into the application of lifetime. Sorry, I couldn't hear.
Curves. I'm curious why the curves are not discrete based on specific absorption quanta, except they're continuous.
Okay, so the excitation was, this experiment was done with a source that was continuous. Specifically is a lamp in the instrument. So you keep sending photons onto the system, and so you record all the photons that are emitted. So as you send photons, it's a statistical process because you have billion trillions of molecules that are being excited, but at the same time you have billion trillions of photons of molecules that emit photons, and so you always acquire photons in a steady state experiment.
I think you want to see the absorption and emission spectra and understand that. You want to see the slide with the absorption and emission.
This one? Sorry?
30s and other stuff in the. I'm guessing I'm looking for the ideal emission and absorption spectral lines, but you see a continuous curve.
It has to do with the Jablonski diagram that you need to explain it to.
Oh, the individual levels. So what happens is that the molecules go to an excited level, S1. That electronic level has a lot of tiny vibrational, and then there are rotational levels, but they are all close together. If you go higher, they dribble down to the bottom of the first excited level, and from there the fluorescence occurs. And it occurs to the ground level. The ground level has these vibrational levels. If it hits, let's say, the first vibrational, it trickles down again to the ground state. But we see the photons emitted by the bottom level of the first excited level.
Any other questions?
Yes.
I have a question. You showed this slide where you measure light pulses in this blue LED, and the excitation pulse length was rather long, I would say. Do you convolve your data, or do you do other tricks other than the convolution?
Right. So all the theory is based on the assumption that we use a very short pulse of light. In practicality, the pulse sources have a finite pulse width. A laser diode is about 70 picoseconds. An LED, like used is almost 1 nanosecond. So there are these mathematical application convolution where we convolute the data acquired with the excitation source.
It seems to work really well, because I mean your excitation time for length is basically the same as the fluorescence lifetime, and it seems to work really well.
Right. You also see it when you measure 77 picoseconds on the rose bengal. It's almost the laser diodes in that case. Yeah.
One more question over there.
So on your example of polarization absorption, and you said going from S0 to S1 versus S0 to S2, you have perpendicularly polarized light that excites that transition. So do you have to know something a priori about the molecule to know that, or is that a general rule for all transitions, like always S0 to S2 is perpendicular?
No, well, different molecules may have the transition moments oriented in different directions. This is let's say, a fortunate case, anthracene, because of the symmetry of the molecules. But if we take tryptophan, is actually the transition moment makes an angle.
So you have to know quite a bit about the fluorophore that you're using in order to interpret these.
Well, usually, correct. Usually you don't know, and you can actually use also these measurements to also separate how the excited states work and infer something about the directions of the transition moments.
Questions? Well, thank you very much.